the anatomy, the histology of the liver is pretty complicated, but it's important to know a little bit about it because it's very closely tied in to the way it works. So I'm going to spend the first few minutes or so talking about some of the anatomy and histology. So overall, here's the liver up in the upper right abdominal quadrant here, and it's connected through a series of ducts. There's the hepatic duct, which leads into a common bile duct, and in most people, the common bile duct, just before it enters into the duodenum, here, the first part of the small intestine, is joined by the pancreatic duct. So there's normally a common entry point of the secretions from the pancreas and from the liver. And this entry point into the duodenum is surrounded by a ring of important smooth muscles, and this is called the sphincter of Oddi. I think that's how you pronounce it, Oddi or Oddi, which can regulate the entry both of pancreatic juice, so I didn't talk about it last time, and also the bile, which is the secretion of the liver, and that's what we're going to be talking about today. Now, just to sort of summarize the functions of the liver that I'm going to be talking about, the liver produces the secretion called bile. This is an exocrine secretion. Exocrine secretions are ones that go into a duct system. And this production of bile by the liver is a continuous process. It's happening all the time. But we don't need to have bile in the small intestine all the time. We really only need it there after we've eaten a meal. Because the bottom line is that bile is important in the handling of fats by the body. Fats that come in the meal, from the stomach up here. Fats are coming down. Nothing really has happened to fats yet. We've got the initiation of carbohydrate digestion by the salivary amylase, initiation of protein digestion by the gastric intestine, but I haven't yet mentioned any fat digesting enzymes. There are some salivary and gastric lipases, but so little seems to be known about them and relatively minor, they're not even mentioned. So essentially, fats at this point are unchanged. And it's to do with the breakdown and the transport and the absorption of fats. That's one of the main reasons why we have bile, and we'll see why a bit later. So, going back to this point of timing, we don't need the bile going into the small intestine all of the time, even though it's being produced. So what do? We divert it into a storage site, the gallbladder. And the gallbladder, it's a bag of basically smooth muscle. It acts as a storage site for the bile in between meals. And then at the appropriate time, and we'll talk about how this happens later on, the gallbladder will contract and it allows the bile to go into the small intestine. So those are just some of the nomenclatures. Now I've put up here really what is a summary of what I said in, I think, the first lecture. But it's, I think, important to emphasize it again here, and that's the blood supply of the liver. It's an unusual blood supply, it's a double blood supply. Remember that the liver receives both oxygen-rich blood from the branch of the aorta called the hepatic artery, and it also receives, in fact, most of its blood, roughly 3/4 of the total blood supply to the liver, comes from the portal vein. And the portal vein is the one into which many of the substances that are absorbed from the small intestine, they pass into the portal vein, and so they're delivered first to the liver before they go anywhere else. So the liver has first sight, if you like, because first go at dealing with substances that are absorbed from largely from the small intestine. So it receives an oxygen-rich supply and also a nutrient-rich supply. And as we'll see, these will mix together within the liver. And then drainage from the liver, the hepatic vein, which in turn drains into the inferior vena cava, and so goes back to the heart. Now, if you take a piece of liver, buy a piece of liver from butchers and take it home, slice it up, look at it, even with the naked eye, a very low power magnifying glass, you can probably see a structure which looks a bit like this at the top of figure number 43. There are these units, these are the basic units of the liver. These are called hepatic lobules, and they're a couple of millimeters in diameter. And what you would see at that magnification would be just these roughly sort of circular, but in fact six-sided structures, and you might see a little sort of dot in the middle, and radiating from that central point are sort of lines, a bit like spokes on a bicycle wheel. And that's all you're going to be able to see there. But if you take one of these hepatic lobules and enlarge it, it's been turned a bit on its side, this is what you would see at a higher magnification. So each hepatic lobule is delineated at its borders, at the points, these 6 points around its hexagonal shape, by a cluster of 3 tubes, 3 vessels. 2 of these are blood vessels. One of them is a branch of a hepatic artery. So when the hepatic artery comes to the liver, it breaks up into smaller vessels, arterioles, and then branches of those and so on. And one of those, the one I've done here in red, ends up in this little cluster here. These are called portal or hepatic triads, or tracts. Triad referring to the fact that there's 3 structures there. So one of them is a branch of the hepatic artery bringing oxygen-rich blood. The second one is dark, it's almost black, sort of purpley one here, is a branch of the portal vein coming in here. So these are both bringing blood to this hepatic lobule, whereas the third member of the triad, the one in green, is a duct of a branch of a bile duct. And this carries the secretion of the liver, the bile, away from the liver. Now, the next figure is going to look at this in a bit more detail. Oh, in the middle is a seminal vein. The next figure is going to, if this is a bit like a cake, and you take a wedge out of this cake like that, cut a slice of the cake, this is what you would be looking at in the next figure. So on the left-hand side will be a portion of the central vein, and on the right-hand side is this portal triad. And in between the two, you've got the main cell type in the liver, the hepatocytes. These ones I've done here in green. So let's just look at this again. You have the branch of the hepatic artery, the branch of the portal vein, and these are both bringing blood, oxygen-rich and nutrient-rich respectively, to the liver, to this lobule, and then you've got branches of branches, and these enter into these spaces which I haven't covered, called sinusoids. And you're looking at them here where they've been sort of cut in half, but in reality they'd be roughly circular, a continuous shape, so that the blood from both the artery and the vein, portal vein branch mix together within these sinusoids. And these sinusoids carry this mixed blood from the periphery, the outside of the hepatic lobule, towards the central vein, and eventually this mixed blood will drain into that central vein. The characteristic of these sinusoids, they're sort of capillary-like structures, but they do show some important differences. There's fenestrations in the wall of the sinusoids, and there's no basement membrane. And those 2 characteristics make it easier for substances to move between the blood and the hepatocytes, which are adjacent to the sinusoid. The other thing to note here is these, the ones I've done in yellow, these bile canaliculus. Canaliculus in the singular, canaliculi in the plural. These are drainage channels for the bile that is produced by the hepatocytes to drain, first of all, into the bile ductule, part of the triad, and then those bile ductules in turn will drain into larger and larger bile ducts, until eventually you've got the one, the main one that leaves the liver carrying the bile ultimately into the duodenum. Notice that these canaliculi are blind-ended. They do not connect with the central vein. So what is a canaliculus? I call it a drainage channel. And what it actually is a space between adjacent hepatocytes. So I'm just going to take a couple of these hepatocytes and show you how these canaliculi are formed. Here's part of one hepatocyte, and here's its neighbor. And what happens is that there are these invaginations in one of the membrane of the hepatocyte, and these line up with the similar invagination in the neighboring cell. And it's this space here that forms the canaliculus. And there are tight junctions here which close it off so that the stuff is flowing through. It doesn't just disperse everywhere. It flows down the length of the canaliculus. And we'll see this again in more detail later on. These are small structures, about a micrometer across. So it's actually quite difficult to do studies on what's going on in the transport systems and so on here and collecting samples, because it is a very small space. But this particular part of the hepatocyte membrane that forms the canaliculus, this is equivalent to or considered to be the apical membrane of this cell, the front door membrane, whereas the other one everywhere else around is the basolateral membrane. So when we talk a little bit about transport mechanisms, I'll be distinguishing between the apical ones, which allow things to move into the bile, and the basolateral one, which allows substances to be taken up from the blood and the sinusoid. We'll come back to that. So I hope that sort of gives you some sort of picture in your mind of the structure, and then what I'm going to be looking at is the relationship of that structure to the function. So what are the functions? Well, I've already told you the main one, and in fact the only one really that I'm going to be talking about here, the role of the liver as an exocrine gland producing bile. The story, of course, doesn't stop there, and you should at least be aware of some of the other functions of the liver. Number two, metabolism and storage of nutrients. There are so many processes going on there. Because the liver is seeing as the recipient of the uptake, the absorption of the end products of digestion from the small intestine, it sees them first. And it's a site where it can store, it can convert one thing to another thing, it can convert the other thing back to the first thing. It can do so many different things. And I was saying here, this essentially is what you would or do learn in biochemistry. It really is sort of a whole course or a whole series of courses just to talk about number two. So I'm not going to. Just to remind you that the liver plays a role in producing proteins, for example, as I did talk about briefly last term. A lot of the blood clotting factors are produced in the liver. Number four, inactivation, detoxification of various substances. I think Dr. Harvey in his endocrine lectures may talk a little bit about breakdown of hormones. Activation again, some hormones, and so on. And then the last one, that's the blood filter. Those sinusoids that I showed you have got these rather special cells called Kupffer cells in them. They are macrophages And they're supposed to be, or not supposed to, they are important in a defense mechanism. So if, for example, bacteria pass from the gut and into the blood draining the gut, and they're heading towards the liver, this is a way of stopping them from draining where these macrophages can play a role in getting rid of things like bacteria. So number one, the bile. What is it? The usual questions. What is it? How is it produced? What does it do? How is it regulated? And so on. So the functions of bile, figure 46, I've already alluded to the first one. It's necessary for digestion and absorption of fats. But I should immediately add there, it's down at the very bottom, but stick it right at the beginning too, that there are no digestive enzymes in bile. So although it plays a role in digestion, it has to be something more indirect. It doesn't have fat digesting enzymes in it. So this is unlike other exocrine secretions we've looked at. Saliva's got enzymes, gastric juice, pancreas, lots of enzymes. The bile does not. Okay? And it has a role in excretion of waste products. So these are the ones we're going to look at, mainly the first one. So what is bile? Like everything else, it's mostly water and the usual electrolytes, nothing particularly unusual there. It's got sodium, potassium, etc. It's relatively rich in bicarbonate, like pancreatic juice, and it helps in the neutralization of gastric acid. The pancreatic bicarbonate is usually considered to be the major neutralizer, but this is certainly helping with that. Some of the more characteristic components are these ones down here, and really the major one are a group of molecules called bile acids or bile salts. Now, I have to admit that my chemistry courses were taken so many years ago that I've forgotten 99% of it. But I do remember that acids and salts are not the same thing. And yet I, and in fact quite a lot of people, do tend to use these terms interchangeably here. So bile acids, bile salts, we know they're different things, I tend to use those interchangeably. So what are they? Cholesterol is a starting material for the synthesis of bile acids. And within the liver, the cholesterol makes what are called primary bile acids. The first one's made. The main ones are things called cholic and ketodeoxycholic acid. They're made in the hepatocytes. Hepatocytes, the main liver cells, produce these from cholesterol, and then you have potential problems. And one needs to understand a little bit about the structure of these bile acids or bile salts to see why there's a problem. They're made from cholesterol. The bile acid molecule is what is called amphipathic, which means that it is both water and fat soluble, essentially. And at its very simplest, you can think of the bile salt molecule looking something like this, and on one side of this molecule are the polar groups, hydroxyl and carboxyl. And these polar groups make this face of the molecule essentially water soluble, whereas the other face, which doesn't have them, is not water soluble, but it is fat soluble. So these are two-faced molecules. One side, which is happy in a watery and aqueous environment, and the other side, which is happy in a fatty environment. And this property of these bile acids, these bile salts, is extremely important and plays a large role in the function of bile to do with fat digestion and absorption. Okay. The solubility of these bile acids is increased when they're conjugated with amino acids, usually glycine or taurine, and then they can form salts with sodium on any other thing. These bile acids, as we'll see, travel into the small intestine. They go down to the furthest part of the small intestine, which is the ileum, or maybe even into the colon, and then bacteria present there can convert them to secondary bile acids, deoxycholic, calcium-cholic, and lithocholic, which comes from the quinodeoxycholic. Okay, now these bile acids, when they're present in low concentrations in the bile, really have nothing to do with each other. They're individuals floating around as individuals. But as the concentration of bile acids goes up, it reaches a critical concentration, and then they start forming clusters or micelles. And a sort of rough approximation of what a micelle looks like is shown in this figure, which you don't have, but you can probably see in various books, or you can see in the book. So here around the outside are the bile salt molecules with their hydrophilic groups facing outwards, because around them is the watery environment of the bile, of the small intestine lumen, and the fatty face is facing inwards. So what this creates is an internal environment which is a very favorable microenvironment for fat-soluble molecules. So you can maybe, looking ahead, you can see, well, here's a role with the bile, specifically the bile salts can play, is in transporting fat-soluble molecules. This function is aided by phospholipids, which are also present, number 3 here, phospholipids such as lecithin, or known as phosphatidylcholine, also present in bile. And these also have they're also amphipathic, their heads are water-soluble, the tails are fat-soluble, so they potentiate the ability of the bile salts to create this internal microenvironment for other molecules which are poorly water-soluble. For example, cholesterol itself, which is found in bile, is very insoluble in water, and it can find itself a happy home in the center of these what are called mixed micelles. And as we'll see later, other fat-soluble molecules produced from dietary fats or certain fat-soluble vitamins can also find a home inside these mixed micelles. So their contribution is very important. The other role in excretion, cholesterol is excreted in bile. Bile pigments. Where do bile pigments come from?
Breakdown of red
blood cell breakdown, or more specifically hemoglobin breakdown, gives you bile pigments. And so this has nothing to do with digestion, but it's just another function of bile to get rid of these things, and other things too, drugs and so on. So where does the bile go? How is it handled? What stimulates or inhibits its production, and how does all this fit together? Well, before going into the details of production, you also need to know about something called the enterohepatic circulation. Anytime you come across the word entero, it means that it's got something to do with the intestine, usually the small intestine. Hepatic is to do with the liver. So circulation. Enterohepatic circulation means that something is somehow circulating between the liver, the intestines, or the intestines and the liver. So what's actually involved? Here's the liver. Cholesterol is converted into bile acids, bile salts. These are sent down various duct systems. Between meals, they're stored in the gallbladder. And then after a meal, they are added to the small intestine. So this is now bile going into the small intestine, bile containing specifically the bit we're interested in is the bile acids or the bile salts. These do their job of helping in fat digestion and absorption, and I'm not going to talk about that today, that will come later on when we look at the small intestine itself, but just for now that's what happens. And then the bile acids, the bile salts themselves remain, they're not broken down at this point, they remain within the small intestine, and they travel right down to the far end, the terminal ileum, just before the large intestine. And the terminal ileum is the major site of absorption of the bile acids, bile salts. And there are special transport mechanisms here for taking up these molecules, and they go into the portal vein and back to the liver. There may be, the dotted arrow here is indicating that there may be a small amount that go actually into the beginning of the large intestine and are taken up from there, but the majority from the terminal ileum and back to the liver. And they can then be secreted again into the bile, and round they go again, round and round. So it's a recycling system. And the significance of this enterohepatic circulation, well, what do you think it is? What's the reason for having it? Why do you have any recycling system, really? You do recycle?
Okay, in a very general sense, the point of any recycling system is to save energy.
To save energy, right? Yes.
To save resources.
Save resources, exactly. It's the same thing here. The total amount of bile salts in the body, number of states make the moment, it doesn't really matter, can be recycled, I think, 2 or 3 times in the course of a single meal. So it can be several times over the course of the day. So it's not a perfect recycling system, only no recycling system is. You are going to lose some in the feces, and you will have to make some new ones. But the vast majority are recycled, and so, as we say, you're saving energy, saving resources. So let's look a bit more closely at how this happens. The production, and the secretion, and the regulation of the bile. So how is bile formed? Hinted at this already. It's the hepatocytes, or the liver cell, which is a hepatocyte, produces these bile salts, etc. By etc., I'm talking about the phospholipids and so on, which I'm not going to go into detail on all of these. And then it secretes them into the bile canaliculus. As this bile salt-rich mixture moves down the bile duct system, it's into the canaliculus first, which then goes to the triad, the portal triad, and then into larger and larger ducts. These duct cells add bicarbonate, so it's a somewhat alkaline mixture going down the duct system. And then, as I said, the gallbladder stores the bile between meals. Now, the capacity of the gallbladder is actually quite small, something in the order of 20 to 50 milliliters. The volume of bile that we produce every day is a lot more than that. It's half a litre up to maybe a litre of bile. So obviously, it wouldn't all fit into the gallbladder, even if you only had to take, a third of that total volume, it wouldn't fit into the gallbladder. So what can we do about that? we do. So the function of the gallbladder is not only to store bile, but it also concentrates it. And it does that by transporting sodium out of the gallbladder, so the sodium moves out, active transport, and water follows. So by taking out sodium and then water following, you get about a tenfold concentration of the bile as it sits there between meals in the gallbladder. Okay. Okay, I'll talk about how it is started in a moment. Okay, regulation. I think in order to understand that, I mean, to just remind you again from this figure that the bile salts secreted, stored, secreted, down here, absorbed, back to here, in the portal vein, and then re-secreted. Now to look at that in a bit more detail, in this figure, we're looking at here but at the cellular level, here's a sinusoid which contains a mixture of portal blood and hepatic blood. So and that portal blood contains the bile salts which have been absorbed in the ileum. That's why they're here. They come from the ileum in this enterohepatic circulation. They come in the portal vein to the smaller branches, to the sinusoids, and they're coming back to close to the here's a hepatocyte, another one here. These bile salts, I just said in that previous figure, they go back into the liver and into the bile. How do they get there? They're taken up by some sodium linked transport system. And if anybody really wants more details on these transport systems, I brought a couple of figures here, but it's far too much. But you're welcome to look at it if you want to. I think all you really need to know here is that there's some sodium-dependent system taking them up into the hepatocytes, and there is some ATP requiring pump system which pumps the bile salts into the canaliculus. And then the phospholipids and the cholesterol and all the other things that are present in bile by various transport systems here also enter into the bile. And then off this goes down through the canaliculus, into the bile ducts, and eventually back into the small intestine. Now, because the bile salts are recycled, if it was 100% successful, we wouldn't need to make any new ones. Once you've made your certain number of grams of bile salts, if you are 100% successful in recycling them, you never need to make any more again. That's not the case. You do need to make some, so there is some synthesis from cholesterol. Now, I emphasize that because if you understand that, then you'll understand how bile salts themselves can regulate the secretion of bile. So normally, or up to this point, when I've been talking about regulation of secretion of various juices, I started as I was nervous, hormonal, both, or whatever. Here I'm starting with a constituent of the secretion itself, which is playing a very important role in regulation, and that is the bile salts. So as more bile salts are absorbed from the ileum, you have an increased amount of bile salts here, and that means that you have more bile salts here, and more can be secreted. So in other words, as the bile salt concentration here goes up, it increases secretion of bile salts. What goes in must come out, but it will decrease synthesis, because the cells are saying, no, you're giving us a lot of bile salts, it's what we need, we don't need to make any more. But if the opposite happened, if something goes wrong, say in the ileum, and the absorption of bile salts goes down, then the amount of bile salts returning to these cells also will go down, and that means that you need to increase synthesis in order to try and bring back to the desired level the body's total pool of bile salts. So this is what I've just said is essentially written down there. This sort of inverse relationship through the control, through the level of bile salts, controlling synthesis and secretion in opposite directions. So normally bile salts, of course, are secreted, and then that indirectly, by changing, I suppose, osmotic gradients and so on, is going to favor water flow and so on, and so affect the actual volume of the bile. So this is something that's a bit unusual. Okay, is there any nervous control? I haven't put any down here. If there is, it's minor. I forgot to say that what I'm talking about here is the intestinal phase. Is there a gastric phase? Is there a cephalic phase of bile secretion? Probably, but minor. Tend to sort of forget about those. And certainly the main one is the intestinal phase, because these things are happening in the intestine itself. The other two are more familiar. Secretin. What does secretin do in the pancreas? Secretin is released when there's acid in the duodenum. Secretin released from the duodenum goes to the pancreatic, which cells in the pancreas does it go to mainly around duct cells, and they increase their production of bicarbonate. Similar sort of thing here, secretin acts on the bile duct cells, and again you get more bicarbonate produced. CCK, also from the duodenum, released by the presence of fats, partially digested fats and proteins. It acts on the acinar cells in the pancreas, giving enzymes, giving more digestion. And it also does two important things here. It causes contraction of the gallbladder and relaxation of the sphincter of Oddi. So contraction of the gallbladder and relaxation of this sphincter of Oddi here. And you have to have both of those happening in a coordinated way in order to allow the bile to actually go into the small intestine. So this is the explanation of why the bile doesn't go into the small intestine all the time. When the bile is being made all the time in the liver, comes down here, if you're in between meals, this sphincter of Oddi is kept tonically contracted. So as the bile is coming down here, part of least resistance is to take this side tube off here and go into the black gallbladder here, because this is closed off, you can't go down here in between meals. But when you eat a meal and you start to get the breakdown of fats and proteins, that will stimulate the release of CCK, which then causes this to contract, this sphincter to open up, and so it's going to squeeze it out. And part of least resistance now is down this way, down here. So hormones do play a crucial role, as well as the role played by the bile salts themselves.